Thanks for the introduction. So my name is Olmo van Akker. I'm part of the Meta Research Center in Tilburg University, that's in the Netherlands. And I'll be talking about selective hypothesis reporting in the field of psychology. And it's part of, this project is part of a bigger project in which I'm looking at how well people or researchers abide by their pre-registrations. And this part zooms in on the hypothesis. So are the hypotheses that are presented in pre-registrations, are they consistent with the actual hypotheses that are used in papers? First, I'd like to give a shout out to my collaborators. Usually people do this at the end of the talk. I decided to do it at the beginning because I'm really grateful to them. They helped me out a lot with all kinds of stuff, with data collection and with tweaking the protocols and piloting. So Marcel van Asten, Marjan Bakker and Jotte Wichert are my supervisors, PhD supervisors. Hou Ong and Andreas Toevenbelt are fellow PhD students. And Manon Enting, Mette de Jonge, Francisca Hoefer and Martijn Schoenmakers are research assistants. And they were really instrumental in getting this project off the ground. So on to the research. So I want to start out with Merton's scientific norms. An acronym of those norms is KUDOS. You see there are communism, universalism, disinterestedness, and organized skepticism. That's KUDOS. But even though there is an acronym, I think these are pretty difficult terms. They don't stick with me for some reason. So I just decided to relabel them a little bit. So communism is basically about the fact that science should be for everyone. So it should be open access. All data should be openly available to basically the whole world, to the whole community of scientists. So of course, we now call that open science. Then there's universalism, which I relabel to impersonal science. That's basically about that science. It shouldn't matter who is doing the science, who's presenting the science. It's about the actual science itself. So it doesn't matter if you're a prestigious school professor, or if you're a PhD student, or even whatever, a baker or something. It doesn't matter. The science should be first. Then there's disinterestedness. I relabeled that to altruistic science, although I'm not really happy with that relabel yet. So maybe you know a better term, please let me know. But that's about putting the science first instead of yourself. So of course we know that science, there's also personal motives for researchers. So you want to make a career, maybe you want to find some prestige for your work, you want credit. But disinterestedness says that ideally scientists should strive for truth seeking. So that comes first, and all these selfish motives should come second. Then finally, there's organized skepticism. That's pretty straightforward. That's just you should be critical about the science. So never take anything for granted. Always be mindful of any limitations of research. So this talk is mainly about the third one, disinterestedness. And this quote is a good starting point, I think. Good scientists should be open-minded in the sense that they consider all new evidence, hypotheses, theories, and innovations, even those that challenge or contradict their own interests. So it's not about your own interests, it's about getting to some kind of ground truth, finding the actual, the truth behind all the noise that's out there in the world. That should trump any selfish interest. That's all nice and well, but not a lot of researchers actually do this. Well, to some extent, so researchers often add, drop, or alter study elements when preparing reports for publication. So that means that it don't incorporate all evidence as they should. This is practice called selective reporting. And it's actually quite prevalent across all sciences, basically. This is a project that's currently ongoing, led by Robbie Thibault from the University of Bristol. And he looked at he basically did a systematic review of all papers that studied pre-registration publication discrepancies. So basically, do people report all the things they plan to do, yes or no? And this is the main outcome table. Then on the left, we see a whole list of things consistent between pre-registration and paper. And here we see the most important ones, I think. So this is about the outcomes, the dependent variables. And here you can see clearly that there's a lot of inconsistencies there. So for example, 68% of all DVs are not consistent between pre-registration and paper. So that's pretty shocking. If you're in pre-registration or in a plan, say, okay, I'm going to do this, and then not do it, that's not how it should work. Of course, there are. It's possible that you have to deviate from your plan, but you have to, of course, then be open about this. But this shows that selective reporting is a pretty big issue in science. But then that raises the question, which science it is. So this paper was based mainly on medical research. You see, that's far and away the largest field or discipline where these studies are carried out in. So the social sciences are only 4. So psychology has 3 papers like this, and economics has 1. And the main reason for this, for the prevalence of these kind of studies in medical research, is that clinical trials are required to be publicly registered. So there's clinicaltrial.gov, for example, where clinical researchers should upload the trials before or during their actual study. That's an obligation by the government. So that basically means that there's a whole database of study plans, and it also makes it possible to compare the publications to the accompanying study plans. So this makes it easy to actually do this kind of research. In the social sciences, we don't really have this infrastructure yet, but still there's been some work done on this. And then people often stick to comparing publications to dissertations. So they look at a dissertation and see, okay, well, what's changed between dissertation and the final peer-reviewed publication? And then they look at selective reporting. It's also possible that they compare publications to archived research proposals. So that comes closer to the comparison of a plan and the output. But I think the golden standard here is comparing publications to pre-registrations, because that's in a pre-registration, that's really your plan. You basically promise, okay, I'm going to do this. And that's the most fair comparison, I would say. So Klaassen et al. Is, to my mind, the only only study that did this in psychology. So here you see the main outputs, the main findings. So in the figure, every cell, every block is a pre-registration. Then we have the black ones. In those cases, they could not even access the pre-registration, so they were not able to compare what was in the pre-registration and what was in publication at all. So that's not the way to go. There was also the darker gray cells. Those are pre-registrations that were really succinct, so really basic. So they didn't provide enough detail to properly make the comparison again. Then the light gray ones, those had some deviations, and they didn't disclose them. So that's what we call selective reporting. And there's one paper, one pre-registration that did indeed disclose all deviations. So there was only 1 out of the, let's say, 30 pre-registrations that actually passed the test here. So this really shows that selective reporting is a problem in psychology. These were, by the way, papers in psychological science. So this is a pretty biased sample. That's why I decided to tackle this project in a bigger way, so to say. So I wanted to look at all pre-registrations out there in psychology and do this comparison for hypotheses. So I have 2 data sources. So one is the pre-registration challenge, which you might be familiar with this. This was a challenge where the Open Science Framework, or Center for Open Science, provided $1,000 to everybody, to anyone who pre-registered their study using a certain template and then published the study. And there are now 180 studies published using this challenge. So I decided to include those in the sample, and I also decided to include studies with a pre-registration badge. So this is an initiative that provides badges to different studies. So you also have open data badges and open material badges. This one is for whenever you pre-register your study to a certain extent, then you get this badge. So these are all also published pre-registered studies. So I combined these, and that led to 424 pre-registered studies. And I only filtered out the psychology ones, so that left me with 329. Then I have some exclusion criteria. For example, well, obviously studies without a pre-registration, where they didn't link to it or where we couldn't find any. Those were, of course, excluded, but also registered reports. So that's pre-registrations that are peer-reviewed before the data is collected. So that's kind of a different system. And because it's kind of a different system, I decided to exclude those as well. And well, in that little box, you see the other exclusion criteria. And in the end, we were left with 613 studies out of those papers. Yeah, and then we excluded those studies, and then we were left with 484 studies in the final sample. So it's quite a lot of studies to look at. And that's, of course, why I'm also grateful to the people that I mentioned in the beginning, because I couldn't have done it myself. Okay, so we have these 484 studies that are pre-registered. So what are we going to do exactly? We're going to look at several types of selective reporting, and we're focusing on the hypothesis, because the hypothesis is the essential element of research, in my view, at least confirmatory research. So we're going to look at changes in the number of hypotheses between pre-registration and paper. So it could be that there are 3 hypotheses in a pre-registration, but that only 2 of them are presented in the paper. In that case, there will be 1 omitted hypothesis, so 1 is not presented in the paper. There's also added hypotheses. That's when you pre-register 3 hypotheses, but then add 6, so you present in the paper, you present 9. It's also an option. We also looked at hypotheses with a status change. So sometimes this actually mainly goes on in medicine, but also to some extent in psychology, that researchers label their pre-registrations as primary or secondary. And of course, what then can happen is if you label a hypothesis primary in the pre-registration, then maybe you demote it to secondary in the paper, maybe because it wasn't significant. That could be a reason. And the reverse thing could happen as well, of course. So promoted and demoted hypotheses. And finally, we looked at changed hypotheses. So these are, this is based on the direction of the hypothesis. So if I say in the pre-registration, A will be larger than B, and if I then in the paper say, okay, yes, as we predicted, B was larger than A, then I'm reversing the order, and that means that the hypothesis changed. So these are the 5 types of selective reporting, selective hypothesis reporting we looked at. Now I'm going to go into the methodology. So how did we look at this? And well, we came across this pretty basic question. So what actually is an hypothesis? That's pretty crucial if you want to identify hypotheses. That proved to be pretty complex, actually. And then a second question was how to identify hypotheses in a reproducible manner. So we want our study to be reproducible. Others should be able to assess whether we've done sensible stuff. So we should have a protocol that's transparent and that people can just assess whether we abided by this protocol. And the protocol looked like this. So what we did was first, we searched for a separate hypothesis section in the pre-registration, for example. And then we looked for some keywords. So these were, for example, hypothesis, investigate, predict, examine. So because it could be that hypotheses are not in a separate section, but in the running text. And then we have to filter them, and we use that based on these keywords. And then we included predictions regarding relationships between 2 or more variables, or when there was only 1 variable, it could also be the case, like a univariate hypothesis. But it had to be a prediction, for it to be an hypothesis. So some stumbling blocks along the way. So what if an hypothesis is phrased in multiple ways? So what we sometimes saw was a conceptual hypothesis. So for example, we expect an association between extroversion and IQ, and operational hypothesis. So we expect an association between scores on this scale and that scale. So that's more in this case, the variables are operationalized already. So sometimes these are both presented in the registration or the paper, and we decided to only take the operational version here. And then there could of course be multiple operational hypotheses. They can test a construct using 2 measures, for example. And then we said, okay, well, each of those measures would entail a different hypothesis. So in this case, we would have an hypothesis involving the record intelligence test and a hypothesis involving the vexillary intelligence skill. Then another complication. Operationalizations are often in different sections. So it's not always that they say, okay, yeah, we expect A and B, A to lead to B, but then further on in the method section, they sometimes say, okay, and B, we measure that using X, Y, and Z. So we had to a little bit copy paste stuff together to actually come to the actual hypothesis itself. So that was a challenge, and the same holds for control variables, because we decided that a hypothesis A without control variables is a different hypothesis than an hypothesis with 2 control variables. So we also had to look at that. And this was our protocol. So this is what the coders have to look at. So this is all kinds of exemptions are listed here. But this goes to show that it's pretty difficult to identify a hypothesis in a reproducible manner. You cannot just subjectively say, hey, okay, pick out hypotheses in this pre-registration, because that's subjective. So to make it more objective, this is our protocol. Okay, what did we encounter then during this phase of the project? A lot of double-barreled hypotheses. So this is an example of that. So if you look at the last sentence, that's the hypothesis. So hypothesis 1. But then that's basically way more than one hypothesis. So it says extraversion, agreeableness, conscientiousness, and emotional stability are associated with subsequent ranked order increases in aggregate levels of daily positive effect and relationship support from child and partner over 1 year. So that's a mouthful. And they just labeled this as hypothesis 1. So what we did was we included all bivariate hypotheses and then the combined hypothesis. So in this case, we cut all these things in parts, basically. So we have, in IVs, we have extroversion, agreeableness, conscientiousness, and emotional stability. And in DVs, we have positive effect, relationship support from child, and relationship support from partner. So that's 3 DVs. So that's 4 times 3 is 12 bivariate hypotheses. And then there's the combined hypotheses. So one of them is, for example, extroversion, agreeableness, conscientiousness, and emotional stability altogether. All of them are linked to positive effects. And of course, you then have 3 of those as well. So this again goes to show that they say it's only one hypothesis, but it's actually way more than that if you're precise. Another type of hypotheses that we encountered were the incomprehensible hypotheses. So if you look at this, this is from a pre-registration, and behind 1, 2, and 3 are the hypotheses. Okay, I have no idea what's going on there. Luckily, there's a key that says, okay, E is the experience condition, for example, and P is the probability condition. But still, somewhere else, they say, okay, we used experimental task from this experiment 3. So we have no idea what the experience condition is or the probability condition. They just refer to some other experiment that they don't go into. So sometimes really have to dig deep to find an hypothesis and be able to comprehend it. So we decided to add as much information as possible, comments in brackets, so that other coders could work from there. The third one is hidden hypotheses. So underlined, you see here that they refer to a table or another file in which they pose their hypotheses. So, okay, we looked at that file, of course, that looked like this. So from somewhere in this field of data, we had to look at the hypotheses. So those are here. So top left, you see that subjective well-being should be related to extraversion. That's a positive relationship. Okay, that's pretty hard to get, I would say. But then it gets harder if you see that this is the whole table that they are presenting. So they just say, okay, the hypotheses are in this table. Here's the table, and then we have to dig deep and look at the table ourselves. So it was quite challenging to find some hypotheses as well. That being said, I think we managed to do that. So having that out of the way, we start to assess the prevalence of selective reporting. So first we looked at omitted hypotheses. And here we had a strict assessment and a lenient assessment. So the strict one was that the hypothesis that was pre-registered should be findable in the introduction or in the method section. So they should say, we expect X to lead to Y, for example. So that's because that's an APA standard. You should, in the introduction, you should pose your hypothesis. And then there's a lenient one as well. And that says, okay, if they test it, then it's already fine. So they don't have to mention it in its introduction. As long as they test the hypothesis, that's fine. So two ways of going about this. Then added hypotheses, we basically decided to look at the results section, and any statistical test result that was not related to any pre-registered hypothesis was counted as an added hypothesis. Only there wasn't the case if they explicitly stated, okay, this is an exploratory hypothesis, or this is an unplanned hypothesis. Of course, they can do that as much as they like. But if I don't mention it, then people tend to it's a confirmatory hypothesis and also interpret it that way. And that's not how what we want. So we labeled those added hypotheses. Then there are hypotheses with a status change. This was pretty straightforward. So we looked at keywords again. For example, key or main or primary for primary hypotheses, and secondary or additional for secondary hypotheses. And finally, we looked at changed hypotheses. So we looked at whether an hypothesis is directional, for example, A is larger than B, whether it's a non-directional, for example, A is unequal to B, or where it's a null hypothesis, A equals B. And if there were any shifts in between these categories from peer registration to paper, then it constitutes a changed hypothesis. So that was our methodology. So now to the preliminary results. We haven't finished data collection yet. We have now collected data for 48 pre-registration study pairs. So that's about 10% of data. And here you see a table of the number of hypotheses per pair. So there were 5 studies with one hypothesis. There were 5 studies with 2 hypotheses, and so on. And the median number for a study, median number of hypotheses for a study was 3.5, either 3 or 4 here. And the total hypotheses coded was 332. So changes in the number of hypotheses. So what we found was that about half of the pre-registered hypotheses were omitted. And I thought it was quite shocking, actually. You can see that in the table as well. This holds for both the strict method that I just described as well as the lenient method. You see that about 50% of the hypotheses that were mentioned in pre-registrations could not be found in the accompanying paper. So that's, I would say that's quite a lot. And if you look at added hypotheses, about half the studies that we looked at with pre-registered hypotheses also added hypotheses, and those hypotheses, of course, were not exploratory, or they didn't explicitly say these are exploratory hypotheses. They just were added there without any disclaimer. So that was in about 50% of the studies. So a lot of changes in the number of hypotheses. So a lot of selective reporting in this area. Then hypotheses with a status change. So this is from primary to secondary or vice versa. So we see the yellow circle constitutes hypotheses with primary importance in the pre-registration. So of those, 7 were omitted, so you can find them in the paper at all. And then the rest, so 23, the authors in the paper didn't specify any importance. So in the pre-registration, they said, okay, this is a primary hypothesis, it's really important. And in the they said nothing about this, they just presented it. Also, no hypotheses were strictly demoted, so none of them in a pre-registration that were primary were secondary in the paper. But also, of course, none of them were labeled both in primary in the paper and in the pre-registration.
Similar findings. Interject with a question from the chat.
Sure.
Don Moore asks, did you consider supplementary materials where authors may have reported results that were less interesting because they were null or turned out to be less relevant to the direction the final paper took?
Yeah, so we were pretty strict in this sense. So if there is a pre-registered hypothesis, we think it should be presented in the paper in the running text, basically. So if you have pre-registered an hypothesis, then you should say something about it in the results. So if the pre-register A is larger than B, you have to test it. So maybe you didn't find a statistically significant result, okay, but you shouldn't hide it away somewhere in an appendix. So in that case, we then labeled it as omitted. But maybe we can discuss later whether that's sensible.
And then maybe just one final question from Lars Wilhuber to wrap things up since we're running on time here. He's asking, how does the change in the hypothesis decompose if you drop all the decomposed hypothesis? And then a follow-up question, what do we know about how researchers perceive the distinction between primary and secondary hypotheses?
The latter is pretty vague. So in biomedicine, that's a more clear cut, I think. Then a lot of templates, like protocols, actually say, okay, what's your primary outcome measure and what's your secondary outcome measure? So it's more formalized in that sense. In psychology, they just say, okay, this is primary and this is secondary, and they are pretty vague about this. What was the first question again?
So it reads, how does the change in
so, if I can summarize it, you coded composite hypothesis into multiple singles plus the composite hypothesis, and then checked whether they were present. From the author's perspective, they don't see that as 16 hypotheses, they see 1. So if you just restricted yourself to how they would originally have been coded, how then does your statistic about they show up in the paper and all those kinds of things show up? Because why would I ever report on a binary hypothesis that never formulated is, I think, a valid question from the perspective of the researcher.
That's fair. Well, first we looked at the statistical analysis plan as well. So a lot of times when an hypothesis was that big, as the example I just gave, then we could deduce from the statistical analysis they were planning what they would do. So whether they would do bivariate tests or whether they would do an all-encompassing test. So a lot of times we were able to find out the intention of the authors. Sometimes we weren't, and then we decided to do it like this because basically to kind of punish vagueness in a pre-registration. So if we don't know what they're going to plan, what they plan to do, what they intend to do, then we can also not assess whether they did what they planned out to do, if you get what I mean. So if their pre-registered hypothesis is ambiguous, we have no chance but to interpret it in a way that both options are equally valid. So we decided to include both options as well in our.
I guess it matters for the count. I don't dispute the idea to break it into more precise components, but if I only registered one vague, then I can only test one vague. I'm never going to test 2 because that would be inconsistent. And so you're inflating to some extent the number of hypotheses that are testable when the author didn't even perceive it as that. So at least as a bound on how consistent authors are within their own framework, not your framework, which they didn't know that you would apply, it would be useful to have that other statistic as well.
Yes, yeah, I agree with that.
I think that's a very important question to discuss, but I'm afraid we're out of time and we're gonna need to move on with the next presentation. So Olmo, if you don't mind staying in this room, and maybe you can discuss this question with Lars in the chat.
Sure.
But yeah, thanks for this great presentation. And I'm sorry that we couldn't get through I think all the materials that you prepared.
I was nearly done.
If you guys, okay, awesome. And I pasted a link to the slides if people want to review them on their own time.